Today we're going to cover respiratory infections. The first part is looking at the causes, so viral, bacterial, for example, the pathophysiology of those respiratory tract infections, before comparing the different types of influenza virus. And as you'll be aware, the common cold generally affects the upper respiratory tract, and the influenza virus generally infects the lower respiratory tract, as well as the upper respiratory tract. So therefore, respiratory tract infections are classified as either upper and or lower according to the anatomical location of the infection. The upper ones tend to be a lot less severe than a lower respiratory tract infection. An upper respiratory tract infection can of course potentially spread to the lower respiratory system. Remember, in your lower respiratory system, that's where you have your alveoli and that's where you're having gas exchange occurring. And so if you're getting infections and inflammation, that's reducing gas exchange, and therefore you're having to breathe harder to get the same amount of oxygen into your system and the same amount of CO2 out again. And as you can see from this diagram, there are lots of potential infections. So you've got sinusitis, rhinitis, epiglottitis, laryngitis, if you're thinking about the upper respiratory system. If you're looking at the lower, you're thinking about TB, you're thinking of bronchitis, bronchiolitis, pneumonia, influenza, some whooping cough. So lots of potential diseases and inflammatory responses that occur in different parts of your respiratory tract. But of course, the most common pathogens are your viruses and your bacteria. And of course, the most common of all of them, the upper respiratory tract infection, is the common cold. And that's often by the virus rhinovirus. And this virus, or hundreds of viruses actually, hundreds of rhinoviruses, they're continuously mutating, which is why there aren't any effective vaccines against the common cold. And chronic lower respiratory tract infections are the 5th biggest killer in Australia, with just under 8,000 deaths in Australia in 2015. And it's often due to a streptococcus pneumoniae infection. So respiratory viruses, though, they have a significant role to play in exacerbating those people with underlying respiratory illnesses, whether that's asthma or COPD, for example. And it increases the hospitalization rate. So there are over 61,000 hospitalizations due to influenza virus and pneumonia infections. And like we said, the most common pathogens are viruses and are bacteria. And so you can see a nice image of an influenza virus up there and a nice picture of streptococcus pneumoniae bacteria. And your hospital-acquired pneumonia infections cost Australia in excess of $500 million every year. So when we're thinking about the infections, after you get inoculation, and therefore invasion by your viral infections, such as your rhinovirus, and your bacterial ones, such as your streptococcus pneumoniae pathogens, there are a number of things that occur. They can obviously be spread through hand-to-hand contact with infected secretions, through droplet infections and aerosols, and like I said, lots of these things spread in the hospital environment, hence the cleaner environment that we need to live in, within or work in the hospital environment. But of course, the invading pathogen has to overcome the upper respiratory tract defences in order to become established within the mucosal tissues themselves. And we have lots of defences in place to try and fight that. So we've got our non-specific defences, such as our mucus trapping. So the mucus that our respiratory tract produces is very sticky, and lots of viruses and bacteria will stick to that, and then we'll obviously remove those through the mucociliary escalator into our mouth, and then we swallow it and it goes into the stomach. You have the white blood cells, which are involved in phagocytosis, so eating up bacteria and infected cells. You've got air turbulence leading to coughing and sneezing, removing the bacteria and removing the viruses. And then you've got your specific immune defences, such as your antibody production against specific pathogens and your T killer cells, which go along to kill bacteria and kill virally infected cells. So what do the antibodies do? Well, as you'll know from your immune system lectures, they target pathogens and infected cells for destruction by these T killer cells, or for eating by phagocytes. So what's the pathophysiology of the upper respiratory tract infections? Well, it's got many mechanisms that lead to the many common symptoms. So you get an initial infection, for example, by rhinovirus. And like I said, there are over 100 different types of rhinovirus. They enter your nasal passage and infect your epithelial cells. And that starts to lead to airway irritations, which leads to your sore throat and your sneezing. The airway irritations and the damage to the epithelial cells releases a lot of chemicals, leading to bronchoconstriction, leads to mucus secretion, both of which lead to a productive cough. Mucus secretion, of course, leads to rhinorrhea, which is your runny nose, and the infection of the epithelium cell. During an infection response, they become inflamed. This leads to blood vessel dilation because of the release of inflammatory mediators, increases your blood vessel permeability, leading to edema and obstruction of your nasal passages. And furthermore, you can get fluid passing into the tissues in the form of an exudate, which again goes on to lead to rhinorrhea. Remembering, of course, that an exudate has got a high protein content. So now we're going to move on to lower respiratory tract infections. And the causes include influenza virus, pertussis, bacterium such as tuberculosis, for example, Mycobacterium tuberculosis. And pneumonia itself is inflammation of lung tissue, and that inflammation results in altered gas exchange. And it can be caused by lots of different things, same as before: bacteria, viruses, fungi, non-infectious agents, irritants, aspiration of gastric contents as well. But 50% are caused by a virus, so virus is very, very common. So if you get infected by a pathogen, such as your virus, again leads to inflammation, can lead to alveolar edema and exudate formation. And remember, if you've got edema and fluid forming in your alveoli, that's not particularly a good thing because they're filled with fluid, and that can include the exudated blood cells and the bacteria, of course, if it's a bacterial infection. And that means you get lung tissue consolidation. And all that means is the tissue contains, instead of gas, it contains liquid. And actually it must be present to actually diagnose pneumonia. So if you look at the mortality stats in Australia for pneumonia in recent times, well, it's 10 years ago now, 2000 to 2009, you can see that as a proportion of total population, you can see that generally females have a greater level of infection than males do. And that's thought to be because the male hormones are more protective than the female hormones. And so therefore more females die from pneumonia every year than males do. It tends to be around about 1,500 females die compared to about just over 1,000 males. So whilst Australia has got relatively few deaths and that's declining all of the time, some countries have got a lot more. So, for example, some countries got 33% mortality rates in children under 5, which is a huge number of people who can potentially die. So, pneumonia infection leads to an increased temperature, a productive cough, and chest and pain fever. And remember what a productive cough means. That just means you've got mucus and sputum or phlegm present. And there are lots of complications of lower respiratory tract infections, such as acute respiratory distress syndrome. And that's just where you have inflamed lung tissue with fluid in the alveoli, which obviously goes on to lead to shortness of breath, rapid breathing, and a bluish skin as you're failing to get as much oxygen into your system as you would like. It can lead to lung abscesses. And really, they're just a type of liquefactive necrosis or cell death of the lung tissue, and therefore formation of cavities greater than 2 centimeters. And these cavities, they contain necrotic, so dead cell debris, and fluid such as pus. And that's all caused by microbial infections. It can lead to respiratory failure, which may require a breathing ventilator, and it can lead to sepsis, potentially leading to organ failure. And there are lots of treatments, of course, and they include things like oxygen, bed rest and fluid intake, analgesic to relieve chest pain, and like we said, ventilation in extreme cases. And you can also get vaccinations against influenza virus and pneumococcal pneumoniae. And people may be susceptible individuals are vaccinated against influenza virus every year. So people with severe asthma, for example, or COPD, they will become vaccinated every year. So a little question for you. So have a little read through this question and then try and answer the questions, true or false in each case. So press pause. Okay, so which of the following are correct regarding respiratory tract infections? Rhinoviruses and TB are common causes of lower respiratory tract infections. That's incorrect. TB is a common or relatively common cause of lower respiratory tract infections. Rhinovirus is, of course, the upper respiratory tract, one of the major causes of common cold. Infection with invading pathogens damages epithelial cells lining the respiratory tract, leading to inflammation and blood vessels becoming more permeable. That's correct. So fluid leaves the blood, leading to exudate formation. Increased blood vessel permeability causes fluid to leave tissues and enter the bloodstream, causing edema and potential blockages. That's incorrect. Fluid leaves the blood into the tissues, leading to edema and potential blockage of nasal passage, for example. Number 4, lower respiratory tract infections may cause pneumonia due to increased blood vessel permeability and resulting exudate formation in alveoli. That's correct. And number 5, fluid within alveoli affects oxygen movement into the blood more than carbon dioxide out of the blood. That is also correct. And that's because carbon dioxide is more soluble in liquid and therefore passes through the fluid better than oxygen. So then what you'll often find is in individuals with inflammation in the alveoli or fluid in the alveoli, oxygen deficiency is more significant than carbon dioxide accumulation within the blood. So what we're going to do now is we're going to just have a look at influenza virus and the different types of influenza virus and why they're such a significant burden on the population. Because they change very rapidly, influenza virus. It's an RNA virus, which means it doesn't have any proof-checking mechanisms, which means it mutates very, very rapidly. And really, every influenza virus that you become or someone becomes infected with, when it multiplies, divides, it produces a slightly different virus every single time, or almost every single time, because of those mutations. Now, some of those mutations are detrimental to the virus, causing it to no longer be able to be productive and infect other individuals. Sometimes it has no significant effect at all, does nothing, doesn't change the amino acid and therefore the protein structure of a variety of proteins. But sometimes it can have a significant effect. So if we look at influenza virus in itself, it's got an incubation period of around about 2 days. Symptoms tend to be a marked fever, headache, photophobia, shivering, dry cough, tiredness, and a dry tickly throat. And that fever can last anywhere from 3 days all the way up to a week or 10 days if you have man flu. Not that I would ever get man flu, of course. You can get secondary bacterial infections, such as Staph aureus, and that's a common secondary bacterial infection. And theoretically, you can diagnose it, so you can detect the antigen, so that's part of a protein, if you like. So influenza antigen can be detected in nasopharyngeal aspirates and throat washings. You can isolate it from nose and throat swabs. So there are 3 main subtypes of influenza virus. There's influenza A, and there are many different serotypes of influenza A. For example, H1N1 or H3N2. And this virus, so influenza A, undergoes both antigenic shift and antigenic drift. And I'll talk about what those two are in a moment. But this is the reason why we can have such big changes in influenza virus, influenza A virus in particular, but also why it can be so pathogenic at times. So birds are a natural host, and the virus can be transmitted to other species, such as domestic poultry, and then obviously onto humans. And this is the most virulent influenza type and causes the most severe disease. We've then got influenza B. There's only one serotype there, so there's no H1N1, HN2, etc., etc. It's just influenza B. Almost exclusively infects humans, and it undergoes antigenic drift only. And it undergoes that antigenic drift and it mutates about 2 or 3 times slower than influenza A. And it's also less severe than influenza A. And finally, we've got influenza C. There's only one serotype here, again, influenza C. It's relatively stable. It mainly infects humans, often children, and it only causes mild disease in children. And it undergoes antigenic drift only. So what do I mean when I'm talking about influenza virus antigenic drift and shift? Well, influenza A virus undergoes both antigenic shift and antigenic drift. It can infect other species, such as birds and pigs, unlike influenza B and C, which only infect humans. And what happens is two strains, for example, from a bird and from a human, if they co-infect a cell, for example, in a pig, you get random packaging of the genetic material, in this case, from the bird and from the human, to create a new viral strain with major modifications. So in this example, the pig is the mixing bowl, and it's got a mixing of an avian influenza with a human influenza to come out with a human influenza, which has got something that we haven't, as humans, seen very much before, or seen at all. So there are major, major changes. And it's thought that the pig is actually thought to be the mixing bowl, where you get this reassortment going on. And it's most likely to occur in places such as China, where animals and humans live in close proximity. There are times when you have more than just two mixing together. So in this case that I've shown you, you've got the avian and the human mixing with the pig to come out with a human. But actually, sometimes the pigs are also infected with the swine flu, swine influenza, and you get a triple mix. And actually, in one pandemic many, many years ago, there was a mix of four. There was an Asian swine flu that mixed with a regular swine flu and an avian and a human to give a new human, if that makes sense. So huge modifications can potentially go on, to which we have very little immunity because we haven't seen those viral proteins before. And of course, that can therefore go on to lead to extensive pandemics about every 10 to 40 years. But of course, as that immunity starts to build to that new subtype, further epidemics and pandemics are more limited. But what is an epidemic and what is a pandemic? So I just want you to quickly define epidemic and pandemic. So an epidemic is where an infection infects many people at the same time. So in other words, more people are affected than would be expected. Whereas a pandemic is basically an epidemic on a global scale. So we talked about antigenic shift, which is basically about the RNA of the influenza virus just being passed into new strains. So you're just mixing the RNA all over the place. Now, antigenic drift is just due to mutations in the virus itself. Okay, and like I said, when you have an influenza infection, that influenza replicates within your cells, in the lungs, and actually you get almost every virus that comes out is slightly different. Okay, so you're getting that antigenic drift happening a lot. But they're only very small changes. They might not affect the protein structure at all. They might have no difference, or it might just kill the virus off. Okay, but each one is slightly different. But because there aren't big changes, you very rarely get an epidemic. So that's why you only get the epidemics due to these new virus strains occurring every 2 to 3 years. And there is still partial immunity because of the cross-reacting antibody induced by previous infections. They're generally not big changes. And of course, there's a vaccine available. And the vaccine for 2018, most people now have the quadrivalent vaccine as opposed to the trivalent vaccine. And as you can see from this, it's just showing you, you have 2 influenza A's and you have 2 influenza B's. And really, when a sample of virus is taken and grown in culture, it is called an isolate. And the particular lab serial number or isolate codes, which is quite arbitrary, but specific to each lab, the animal the isolate was taken from, its location and the year all become part of the strain designation. So hence, influenza A was isolated in Michigan with an isolate code 45, don't worry too much about that. And the year was 2015, and because it's influenza A, it was H1N1. And the second one is influenza A, isolated in Singapore, don't worry about where from, 2016, H3N2. And then you've got influenza B, Brisbane, 2008, and Phuket, 2013. So you can start to see how they're actually designated. So there have been avian influenza outbreaks, and you can understand why now, because we've got that big mixing bowl going on. And there was a recent outbreak of avian influenza because of a strain of H5N1 influenza virus. And when we talk about H1N1, H3N2, H5N1, it's all down to the proteins on the surface of the virus. So H just stands for hemagglutinin, and it's number 5, and N stands for neuraminidase, and this one is number 1. So they're just slightly different proteins. So you can have a H3 there, an N2, whatever it might be. And this bird virus infects humans without undergoing a recombination event in some other animal. In other words, there was a species jump to humans because of drift. Now, this is rare. Normally, the species jump occurs because of shift, antigenic shift, not because of antigenic drift. Now, in this case, the fatality was about 60% in humans. Luckily, as the virus wasn't readily spread from birds to humans or from one human to another, there weren't a huge number of people actually killed. However, it could theoretically mutate or undergo reassortment with another human influenza whilst remaining as virulent, because remember, 60% fatality is quite virulent. So, how do we actually get a pandemic starting in the first place? What are the three conditions that need to be met? Well, you need to have a new influenza A subtype emerges to which we have no or very limited immunity. And that was the case with this H5N1 avian flu. The virus can infect humans, resulting in serious illness. This was also the case with H5N1, remember, with 60% fatality. Human-to-human transmission is efficient. Fortunately, with this one, this was not the case. It was very, very inefficient at human-to-human transmission. Therefore, we really did not get any pandemics or epidemics. But theoretically, you could do, okay, if it was to reassort with another virus that spread a lot better. So there have been some serious past influenza pandemics. So the earliest one that lots of people refer to is 1918 H1N1, which is Spanish influenza, which killed upwards of 40 million people. You've then got some other ones. You've got the Asian flu, the Hong Kong flu. You've got re-emergence of the Spanish flu in 1977. You've got swine flu, which kicked off in 2009. And then you've got the seasonal flu, which remember is often H1N1, H3N2 with your bees. And that kills around about 500,000 people every year. So it's still a significant killer. Death rates, of course, tend to be a lot lower now as antibiotics are available, which are helping to prevent secondary infections. So remember, you get influenza and then you might well get a secondary bacterial infection. But there was some controversy because a few years ago now, well, what's that, 14 years ago, the H1N1 genome was published in the magazine Science. And there was fears that this information could potentially be used for bioterrorism. It doesn't appear to be the case, but there were fears at the time when it was published. So really I want you to just do a little this is a comparison table, and I want you to think about what have you learned about influenza A, B and C? Which is the nastiest? So what's the illness severity? We can just put pluses in there. What's the animal reservoir? So where can it actually multiply? Do we have human pandemics? Do we have human epidemics? And what are the antigenic changes? Is it drift, shift, or both? Okay, so press pause and have a little bit of a think. What can you remember on these different types? So illness severity. Influenza A, lots of pluses, very nasty. B, we're carrying on influenza A. So animal reservoir, yes. Human pandemics, yes. Human epidemics, yes. Antigenic changes, that's both drift, which is your mutations, and shift, where you're getting reassortment of genetic material. Influenza B, illness severity, a little bit nasty, but not too bad, but it is included in the vaccine, of course. Animal reservoir, no, only affects humans. Pandemics, no. Epidemics, yes, you do get more infections because of it, and you get these localized epidemics. Antigenic changes, just drift, so just random mutations. And there's much slower mutations than in influenza A. Finally, influenza C. Illness severity, it's fairly mild and mainly in children. Animal reservoir, no. Pandemics, no. Epidemics, very, very occasional. Antigenic changes, drift, but it's very stable, so it mutates quite slowly.